Next, we have Laura Haynes from the University of Michigan.
Great. Thanks for letting me give this talk today. Before I get too started, I want to acknowledge my co-author, Matt Holding, who's another postdoc in the lab, in David Ginsburg's lab at University of Michigan. And he's an evolutionary biologist by training, so that's really helped to get this project off the ground. So serine protease inhibitors, or SERPINs, are a ubiquitous family of proteins that are found across all kingdoms in life. And in humans, there's over 30 that regulate distinct processes by inhibiting specific serine proteases. So they do this. So all SERPINs share a common fold. The salient feature is that they have a reactive center loop, or RCL, that extends from the central globular portion that contains the amino acid sequence that is the preferred cleavage site of the serine protease that they're targeting. So the serine protease will attempt to cleave this reactive center loop, but it will fail to do so. So before the acyl intermediate can be resolved, RCL inserts itself into that central beta sheet. And now in this mousetrap mechanism, we have an inactive SERPIN and an inactive serine protease. And so the prototypical SERPIN that I'm using in these studies is plasminogen activator inhibitor 1, or PI1, which canonically inhibits UPA and TPA, and essentially it acts as the brakes on the fibrinolytic system, so it slows down the fibrinolysis, or the breakdown of fibrin clots. And so we want to ask the question, can we alter the specificity of PI1 for other serine proteases that aren't involved in this process? So in addition to UPA, can we inhibit other members of the coagulation or fibrinolytic pathways? So we looked at thrombin and coagulation factor 12A, as well as another protease of interest, TMPRSS2. And we wanted to see if the mutations we're introducing that alter specificity would just be in the reactive central loop, or if they'd be distributed globally throughout the protein. So we do this by building a phage display library, and what's really great about these studies is since we're forming a covalent complex between the protease that we're targeting and the PI1 variant, our selection is very simple. And then we can just do an immunoprecipitation of the complex and then continue with our DMS pipeline to figure out the enriched and depleted variants or gain a loss of function. And so what this leaves us with is our specificity fingerprints. So you can see here for UPA, we have variants that obviously maintain the inhibitory capacity of PI1, as well as variants where you lose the inhibitory capacity. And if you look at these other target serine proteases, you can see that a similar yet different profile emerges. So to get at this question of where are these mutations happening, kind of fell out of how we designed the experiments as our initial pass for doing this analysis. So we sequence this using 12 overlapping amplicons, and I'll draw your attention to amplicon 12. That's where that reactive center loop that we think of as the through this dogma of SERPIN biology is where the specificity comes from. And in fact, if we look at these PCA plots generated across those 12 amplicons, we can see that the specificity isn't just in amplicon 12, it's actually distributed across the entire sequence space of PI1. In the variants that we've generated, really showing how important exocyte interactions are, as well as allosteric networks are to regulating the specificity of the protein. And so the final question that we've been looking at with this data is we're saying how far from the variants that inhibit UPA are these variants that inhibit these new target proteases? And so you can see here, the UPA in this three-dimensional PCA plot is shown in green, and then factor 12A it's the least different, whereas TMPRSS2 and thrombin seem to have moved the farthest away in their variant profiles. And so to get at why this is the case, we looked at the evolution of the serine proteases that we're inhibiting. So this is your UPA and TP here on this phylogenetic tree of human serine proteases, and interestingly, or maybe not surprisingly, factor 12A is the most closely related to UPA and TPA. So it does make sense that we wouldn't have drifted that far in the inhibitor space as well. And you can see as you look here, the coagulation, TMPRSS2 is the next most closely related, and thrombin is the most distantly related of the serine proteases that we're inhibiting. So it really shows that when we are trying to evolve these inhibitors, their evolution is really matching the natural evolution that's occurred for these serine proteases. So with that, I would like to acknowledge my laboratory and our funding sources, and I'm happy to take any questions.
I imagine folks have looked at sort of coevolutionary patterns of these, inhibitors and proteases before. Is there a way you could think about sort of designing libraries, on both of these sides of the selection to sort of get at pathways of how that evolves?
Yeah, that's a great question. So actually what we're doing, right, so I think I can answer half your question. I've really been focused on the SERPIN side, the SERPInology side of it. So to get at that question of how those pathways have evolved, we actually have generated a common ancestral variant of Pi1, and we're using that to see if those pathways will match. But I also think this retroevolutionary work of going from the modern proteins and seeing how you could go back to those historic variants is also important to really cover that complete mutational space. So that's how we're trying to cover that. Thank you.